Hello everyone, today I'll be solving chapter 3 of human biology as the 14th edition. This chapter is about cell structure and function. So let's start. Obviously I'm solving the end of chapter questions, the assess yourself part. Number 1, as the size of a cell decreases, the ratio of surface area to volume. So let's see, surface area to volume, its size decreasing. I don't know what is like the shape of the cell, so I will not give a specific formula. However, what is known that if you're doing surface area, it's always r square. If you're doing volume, it's always r cube. Okay, this is the volume and surface and the respective surface area formula. So if you decrease your size, obviously your radius is gonna decrease. I'm talking about the cell, not you, so obviously your cell is gonna decrease. But since the volume it's r cube, your volume will decrease more than your surface area. So in the end, since your volume is decreasing more and since its ratio of surface area over volume, your numerator is increasing while your denominator is decreasing, thus the ratio will increase. Okay, got it? Next question. The cell theory states that all life comes from pre-existing cells. Yes, we believe that we do not know what caused the original cells to basically exist, but it's a fundamental theory that all cells, they come from pre-existing cell division. All life is composed of cell. Yes, cell is the basic unit of life. This can be the definition of a cell. And yeah, option C, cell is the basic unit of life. Yep. So it's option 3, D. All of these are correct. Coming now to question number 3. Prokaryotic cells. So the main difference between prokaryotic and eukaryotic are the membrane bond organelles. Obviously, mitochondria, nucleus, all of these are not present in prokaryotic cells as they tend to have no-membrane bond organelles, and they also tend to be simpler in complexity when compared with an eukaryotic cell. So cytoplasm, so the question, let's read the question. Prokaryotic cell contain all of the following except, keyword except, cytoplasm. All cells have cytoplasm. Plasma membrane, also known as cell surface membrane, all of them have it. DNA, when we say DNA, we refer to the genetic material. All of them have genetic material. Nucleus. Now, imagine this is my cell. When I refer to the nucleus, I mean the nuclear envelope, along with the nucleus and the DNA. All of this structure is called the nucleus. So, as we said right now, is that prokaryotes, they do not have membrane bond organelles. So they do not have a nucleus. Their nucleus is just free. It's like strings like this, like the genetic material DNA is present, and it's present in a region called the nucleoid, but it's not enclosed by any sort of membrane. Coming to question number 4. The endosymbiotic theory explains which of the following. So this is a theory about the cells coming from how we eukaryotic cells have like mitochondria and chloroplast. If you look at them via microscope, you'll find that they have their own ribosomes and their own DNA. So this all explains the origins of the first prokaryotic cells. Actually, we still don't know how we got the first prokaryotic cells. The formation of the plasma membrane, your cell surface membrane, how it's formed, no. Why DNA is the genetic material of all cells? Nope. How eukaryotic cells evolved from prokaryotic cells. As I told you, is that it's why, for example, why we believe that all cells started as prokaryotes, including mitochondria and chloroplast, and like some sort of pressure caused them to evolve into a eukaryotic cell, but they still retained their own DNA and RNA. So believes that eukaryotic cells developed or evolved from prokaryotic cells. Coming to question number 5, which of the following is not part of the fluid mosaic model? So this is the model that represents the cell surface membrane or plasma membrane. The fluid, it stands for like the phospholipids, and the mosaic it's basically describes the pattern of all the proteins, the glycoproteins and glycolipids and all the transport proteins and their arrangement in the plasma membrane. So just right now we mentioned it's the plasma membrane. It contains phospholipids, proteins, transport proteins for active transport and facilitated diffusion, cholesterol helps to maintain the fluidity of the membrane. For example, if it gets too hot, obviously phospholipids are fats. If you put like butter or something on a hot day, it's gonna melt. So your plasma membrane will be like they will just be melty and anything can come in. However, cholesterol actually maintains the fluidity. It will basically, you can say in our example, it will toughen up or solidify the plasma membrane more, and it does the exact opposite when it's high temperature. When it's rigid, it allows it to be fluid, so it just maintains the fluidity during temperature changes. Chromatin, chromatin is actually in the DNA, so it's not part of our plasma membrane. Coming now to question number 6. Facilitated transport differs from diffusion in that facilitated diffusion, so I'm comparing facilitated diffusion and diffusion itself, involves the passive use of a carrier protein. It's true. It's facilitated passive, meaning it's no energy. And yes, we do use a carrier protein. Involves the active, no. If I was comparing with active transport, yeah, but I'm not comparing with active transport. In our in the transport mechanisms of our body, active transport and endocytosis and exocytosis are mainly the ones that use energy. I did a video covering the transport mechanisms, you can check it out. So active, no. Moves a molecule from a low to high concentration. Nope. It actually does the opposite, from a region of high concentration to a low concentration. Anything that uses energy does this. Involves the use of ATPs. Again, ATP is like the energy currency, and we're not using energy. So it's only correct is option A, is that they both the diffusion and facilitated transport differ in the sense that in facilitated I have to use a carrier protein for for movement of polar substances. Coming to question number 7. A cell placed in hypotonic solution. So an easy thing to remember is that hypo, in other words in biology, hypo is less or low. Okay, let's say low. Hyper, more. Okay, hypo, low. Hypotonic, so tonic, it refers to like the solute concentration. So hypotonic, it means that it has a low solute concentration. In other words, it has a high water potential. So you placed your cell, what will happen to it? Since the outside is having a higher water potential or a lower solute concentration, what will happen is that water will move, osmosis will move water from outside to inside your cell. Water enters the cell towards the area of higher solute concentration. Yes, because we said that obviously since that one is hypotonic, your cell, meaning that it has a lower water potential, or also it has a higher solute concentration, it's hypertonic. Let's go now to question number 8. Okay, question number 8 is sort of like in the blanks, or choose the correct answer. So we have Golgi apparatus, nucleus, ribosome, and lysosome. Question number 8, location of the chromatin and nucleus. Listen, right now we discussed this. It's the nucleus B. Organelle, where proteins and lipids from the ER are modified. Modification. Whenever you see this word, it's the Golgi apparatus A. Modification and transport. Golgi apparatus, especially proteins and lipids. Yep. Contains digestive enzymes. This is basically the job of the lysosome, in which its job is to break down damaged and old organelles. So think of it like the cleanup crew. Site of protein synthesis. Again, keyword protein synthesis, ribosome. I feel like if you just you got the keyword, you would get it. Now coming to question number 12. I'm gonna put this a little bit here because it's messing up. The ribosomes on the rough endoplasmic reticulum assemble. So on the rough in ER, I have obviously ribosomes. We said now protein synthesis, so it's proteins. While dash are assembled in the smooth, the function of the smooth endoplasmic reticulum is lipid synthesis and detoxification. So let's focus right now on the first part, lipid synthesis. So the thing that we have is phospholipids. Yes, we didn't specify a certain kind of lipid, it's just they do lipids. And phospholipids basically a lipid in which one of the fatty acid tails it's replaced with the phosphate head. So yeah, proteins and phospholipids. Coming to question number 13. Cytoskeleton of a cell consists of all of the following. So obviously cytoskeleton is basically like this, the structure that gives you the shape of the cell basically. And what is made of? Microtubules. Yes, of course you might have famously known them during cell division during the stages of mitosis and meiosis. So yes, they do give the shape of cytoskeleton, the cell its shape, and they also are involved in the transport of substances in the cell, across the cell, let's say. Actin filaments, yes, these are like fine filaments. Extracellular matrix, intermediate filament. So the cytoskeleton is made up of two types of filaments, actin and intermediate. And even if you do not know that, like, just want you to use like logic, extracellular is something outside of the cell, and I'm asking you about the cytoskeleton of the cell. So obviously it cannot be extracellular matrix, extracellular fluid. Extracellular matrix, it's the thing outside of the cell, and it does consist of filaments. However, it consists of other types of filaments, not these ones. 14. Cilia and flagella are involved in forming junctions between cells. No, that is the job of like these filaments and the plasma membrane, and it's called gap junctions. Establishing the extracellular matrix, not their job. Cell to cell communication, again, the cell to cell communication happens through the junction. Cell movement, yes. Cilia and flagella are like made up of microtubules, and just right now I said that it's like doublets of microtubules around the singlet, and it's especially prevalent like in bacteria. For example, if I draw this is a bacteria, you'll have a cilia and flagella here, so it's just use it to swim and move. Number 15. The active site of an enzyme is identical to any other enzyme. It's identical to that of any other enzyme. Nope. Imagine you have two blocks and they're identical. Okay. Obviously they can't fit each other, which basically negates the idea of an enzyme. It cannot do its job like this. Number 2 is that it is the part of the enzyme where the substrate can fit. So let's say I drew it like this. Obviously I'm not gonna draw them identical. So, and again here I have my substrate. So this is my substrate and this is my active site. Yes, the active site is a place where the substrate can fit, and it does its job by lowering the activation energy. So it is option B. Destroyed during chemical reaction. Nope. Is where the coenzyme binds. No, a coenzyme, it's a non-protein molecules that helps the enzyme do its work. Okay, enzymes accelerate a chemical reaction by, so the mechanism by which enzymes work is that they lower the activation energy that the reaction need. What is the activation energy? It's basically the minimum amount of energy you need to start a reaction. For example, you need 500 joules minimum, let's say, to basically extend your hand. Okay, so 500 would be the activation energy. However, like to move your hand and put it away, you need obviously more than that. So what the enzyme will do, it will lower the 500 to, let's say, 300 or 200, so that the reaction can happen. Which of the following pathway? So this one is, it doesn't increase, it just lowers the activation energy. Okay, now this question is, which of the following pathways produce the greatest amount of ATP? So we have how to make like the aerobic respiration in mitochondria. It has 3 main steps, we can say 4. So the first one is obviously glycolysis. It's in which glucose is formed into, is changed into pyruvate, and this one is anaerobic, meaning it doesn't require oxygen, and it produces 2 ATP, you can say. For every 1 glucose molecule you can get 2 ATP. Then you have the citric acid cycle, in other words it's Krebs cycle. Again, this one happens inside of the matrix in your mitochondria. However, it does not release that much. Again, it just releases like on average 2. Coming to the electron transport chain, which can only occur when you have oxygen in aerobic conditions. So when you have oxygen, what will happen is that, to keep it simple, the electrons will pass through the electron transport chain, and then basically you'll get to pass through ATP synthase. Anything ending in ASE is an enzyme. So ATP synthase, from its name, it's an enzyme that makes ATP. So once your electrons, your hydrogen atoms, your hydrogen ions pass through this, you get a very, very good supply of ATP. Like you can say it's about in the 30s, 30 something ATP you would get. So it is the one that releases the most energy. Fermentation happens when in anaerobic conditions, and we said that in anaerobic conditions the only thing that's happening is glycolysis, so we only get to ATP. 18. Which of the following reactions is aerobic and recycles NAD plus molecules? So I want aerobic, so fermentation, nope. Glycolysis, nope, because they do not require oxygen, so these are not aerobic, they're anaerobic. Recycles NAD plus, so recycling is basically returning it to NAD. Okay, I want to return it to its original form. How am I going to return it? It's returned when, so as you can see here, it's plus, it's missing an electron, right? Returning to our chemistry, whenever something is positive, it means it lost an electron. And I said that the electron transport chains is basically electron moving through it. So an electron transport chain, once the oxygen, once the hydrogen come back, they will be united and you get NADH. So this is the one that recycles NAD plus. Hope you guys understood it very clearly. If you have any questions, drop it in the comments. Thanks for watching.